Good afternoon, good morning, good evening, depends where are you. And now we have an afternoon panel session. Oh no, sorry, a keynote session. We have our second keynote, Professor Zhanfeng. Professor Zhanfeng is the Donald Podlock Professor of Chemical Engineering of University of Oxford. He is also the founding director of the Oxford Center for Tissue Engineering and Bioprocessing, and the founding director of the Oxford Suzhou Center for Advanced Research, Oxsed in short. He was educated in China and then come to United Kingdom, Strathclyde University in 1990, I think, and then become a lecturer in Edinburgh University in 1991, and moved to Oxford in 1994 as a university lecturer, and then become professor of chemical engineering in 2000. He's one of the first few Chinese professor in United Kingdom. And then he became a fellow of the Institute of Chemical Engineering and a fellow of the American Institute of Medical and Biological Engineering. And then in 2013, he was elected as a fellow of the Royal Academy of Engineering. Since the pandemic outbreak, Professor Cui led his team in Oxford and in Suzhou, both in the United Kingdom and in China, to develop the rapid testing facilities, which is lab-free, very suitable for use in a developing country. And this revolutionary development won the President Prize of the Royal Academy of Engineering just a few weeks ago. So it's our great honor to have Professor Zhanfeng here today to talk about his invention. In particularly, he's a culture mind about developing this one for the developing world. Professor Cui, stage is yours.
Okay, well, thank you, Professor Guo. And then one thing you introduced, thank you for the introduction. You introduced so much, but the one thing you forgot to mention is that we are good friends. I think,
okay.
Thank you for inviting. And good afternoon, and in Hong Kong, and good morning in UK. And it's a pleasure actually to share some of our experience with you. I say I actually visited your university once before when you gave Donald Berkeley, and who is the one Chinese novel get the Nobel Prize, and you gave the honorary degree to the Mo Yan. In that ceremony, I attended the ceremony and the dinner, so I actually had a good memories. Right, and let me share my screen with you with a PowerPoint presentation. Okay.
Professor Cui, just to let you know, this is not just to the Baptist University. The conference attended by the people all over the world.
Oh, right, good. I have to be more careful. Everybody in the world, yes. All right. Yes, so but today I actually want to talk two things. One thing is about our test, okay? And the other thing is related to the international collaboration. I think it's actually very important when we're dealing with this pandemic, like facing the whole world, and the international collaboration would be the key. And as Professor Guo introduced me as a director of Oxford Center for Suzhou Center for Advanced Research, Oxsed, I will actually start to introduce this to say a few words about Oxsed. The reason will become clearer, and because actually this is why we do the job is because of Oxsed started this test program. So this is called Oxford Suzhou Center for Advanced Research, right? In Suzhou Industrial Park in SIP. So this is actually the University of Oxford, the first center of science and engineering outside the UK, outside Oxford, the Ring Road, basically. We are all in Oxford. So this is the first thing in Oxford history, 850 years, depending on how you count it, and set up something outside Oxford. So it is actually quite a big step internally, because Oxford is actually very cautious. I don't need to do introduction to Oxford University, as I guess people all use dictionaries of published by Oxford Press, University Press. So I just say a few words about Oxford OSCA. So this is the first center, and we're in Suzhou Industry Park, just outside Shanghai. And so we went there for various reasons. One of the bigger major driver would be the industrial base, because SIP has a very strong industry base. We like to move in our research to the end users. That is one of the main drivers. So it, as you expected, to set up something like this, it takes quite a long time. Actually, we initiated the discussion in 2012, and take about a year. In April 2013, we signed the MOU. That's in our previous Vice Chancellor, Professor Andrew Hamilton. He is now president of New York University of New York. And then it take about 5 years, or more than 5 years. And finally, in 2018, 22nd of November, that's a Thanksgiving Day, we had a grand opening. So our current Vice Chancellor, Professor Louis Richardson, went there and opened that. So it's actually a big event to Oxford, and also to China as well, because Oxford finally landed in China. We were given a 9-store building, and we actually it's an empty building, and take quite some time to refurbish it, because we like to have a forward-looking. When people think about Oxford, the first thing come to mind, we produce prime ministers, we publish dictionaries, but when you say science and technology, seems quite often ignored. So what we like to set up in Suzhou is a showcase in science, technology, and engineering. And also Oxford is not just old, it's also forward-looking. So that's the design concept, and then we refurbish the whole building. And all the PIs are from Oxford University. It's all tenured professors, permanent professors, and that's a PI. So we have 16 PIs in 4 areas: biomedical engineering and healthcare, environmental and biotechnology, nanotechnology and functional materials, quantitative finance. So that's each area we have 4 Oxford professors to lead. And why we choose these areas? Because we have strengths in Oxford. Because because the last thing we want to see is that you go to China. If we don't go, people say Oxford, you people must be very good. But when we get there, they said, oh, you're just a so-so. So we have to protect our reputation. So we only do the area we have strengths. And this also actually fit the SIP environment, because they have a local industry base, or like in finance, is very close to Shanghai. So that is actually the rationale. So we employed more than 50 researchers, postdocs there. Is all employed in China, but with Oxford, the salary is the same as the salary scale is same as Oxford. We have dozens of supporting staff. But the one feature of this center is wholly owned by University of Oxford. So it's not a joint venture. It's actually, basically we operate just like a department in Oxford University. So that is how we set it up. So it is actually a showcase for international collaboration. And then I encourage you, when you go to Suzhou, go to SIP, and pay a visit to us. And because it's just a part of Oxford University, it's just like 6,000 miles away. And so that's that introduction. Now after that, I will say, moving to the test kit program. because Oscar is where the test case program started. So this is how it was started. In later January, it's Chinese New Year time. So we all actually come back because also Chinese New Year, we all plan to come back to China. And so what I forgot to say is I'm actually a director of Oscar. And so I've been through this whole process. I spend probably 3 months every year in China and to run the center. So before Chinese New Year, we all come back to the UK because in holiday, even before the virus spread. So we come back passing through Heathrow because at that time, China is a high alert, right? Everywhere is this virus, the Wuhan got locked down almost. And so we passing through Heathrow Airport, and there's no single question asked about the virus, about this issue. And of course, there's no measurement of temperature. You're just walking through. We said, this is nuts, you see. If one side in China, we are all high alert, and then we're passing there, nothing happened. We said, we need to test the passengers. That's where actually the idea immediately first came. We said, look, we have to do something. So this is on the Chinese New Year Eve. So a few of us, we said, let's have a drink. And we said, we need to do something for China, because at that time, there's nothing outside China. We said that China is going to face this problem, and we're now running back. We're hiding in Oxford. We don't get infected. This is we needed to do something with China because we work there. So we are not a medic. We can't do anything with vaccine. And we said what we could do. So we said let's do something with the test. Let's see, because we're passing airport. If we got it there, we got something we can use for airport. That's how it started. So four of us had this New Year's drink, and then we said we decided to do something. I think the good thing is in Oxford is that we have a lot of good people. So we quickly assembled a team, none of us working on this thing, on IVD or diagnostics. So the key leader is Professor Wei Huang. He's an associate professor in the department. He's an EPSRC fellow in synthetic biology. He's also Oscar PI. And then we have a few postdocs working on synthetic biology, tissue engineering, stem cell therapy, biomaterials, but none of us working on diagnostics. But then we said, this is something we want to do. So let's just do something together. And so we assembled this team. At that time, Oscar was in lockdown because of holiday and the lockdown. So we started to work at Oxford. So when we talked to the university here, each of us, we have labs. We have a much bigger group here in Oxford. So we talked to the head of department and the university. We said that we like to do this, but we are in Oxford. We are not in Suzhou. The university people immediately realized, I think they are far more experienced. They said, look, this is not just for China. This is for the problem of the world. Sooner or later, we all face this. Start as soon as you can. So we got the lab access. We set up the new program in Oxford. So the university, I have to say, is very supportive. And they realized that even at that time, later January, they said, this is not a problem facing China. Is facing everybody. We will face everybody. We'll be facing this problem as well. So we actually get the teams when Oxford locking down, this most of the work done in China. And the problem later is that to get validation because there's no cases in the UK. And then we can't ship things back to China because all the airlines is crowded. So back to what we started. This is actually, this is the initial thought. We said, how we test the passengers at the airports. Okay, so you just come out from the quickest, like say Heathrow Airport, Terminal 5, British Airways. If you landed on Terminal on A, that part is the closest. The quickest that you can get off the plane, get to baggage claim immigration, is at least 30 minutes, okay? So what we really want to do at the airport, of course, is this individual got a virus or not. If that's a key thing. So then depend on the outcome, either go to isolation or you just free to go. So that's why just arbitrary, I pick up the number 30 minutes. So that we need to test in 30 minutes. And then you can't send the sample go to the central lab. So we need to be quick. We need to test on spot. So that's the task. I said, guys, this is my requirement to you. Let's work on something out. So then the question is how? Okay, so what to do, because this is in later January. And then I think this is why we got a few weeks in advance compared to other people, because other people in the West only realized when the problem spread. But we came from China, we were aware of the problem. So we got a few weeks in advance compared to other groups. So we pick up then, in order to get this quick on sports, we did actually a survey of different technologies. Then we finally settled with LAMP. LAMP is loop-mediated isothermal amplification. It's a technology to amplify DNA. And it's not a new technology, actually. It's invented by Japanese scientists 20 years ago. But the reaction is random, is very difficult to control. It causes a lot of false positive. Therefore, it's not widely used in the last 20 years. So you think the advantage, the fast detection of virus would be great, isn't it? But then it's not used much. So but then the standard PCR, conventional PCR, take at least 90 minutes. Even you got a sample, take 90 minutes reaction. So it's not suitable for us. So we said that we do the LAMP, but then we have to modify it. So basically this is all from Professor Huang's work, contribution. So he designed high specific parameter sets, only target this virus, particular virus. And more importantly, he designed the molecular switch to control the reaction. So the reaction is in a controlled manner. This will completely eliminate false positive. Okay, so there's no false positive. And so this is both patented technology now. So I come to say, this is all background thing. But then, so what we have done, what to test? This is come to you heard on the news, particularly in UK at the moment, because the Prime Minister announced this moonshot program, billions of pounds will do screening to do test, but what to test? So it's actually, if you see, there's two types of tests, basically. One is to test whether the individual got a virus and is infectious. This is like, if I organize a party, guests arrived, I said, look, I'm feel uncomfortable whether people got a virus or not. So this is a test that you need to do, and the same as in the airport, right? So there is two types. One is the nucleic acid test. This is a standard PCR to test the virus RNA. Because the RNA can be transcripted, it can be changed to DNA, and the DNA can be amplified. So you can detect a very, very low limit, a small number of the virus. You can amplify it, then you can get accurate detection. Okay, you can get a detection limit, you can detect. So this is actually the standard PCR test. The other, and also our LAMP test. The other test is so-called antigen test. If people may notice that Abbott announced the $5 test for antigen test. This is test the virus specific proteins. Because you detect the proteins, you cannot amplify it. So it's because you do anything, the protein will denature. So therefore, although it's fast, but it's less accurate. If you can only have a lot of proteins, you can have reliable detection. But the other type of test is antibody test. This is actually blood test to get antibody. But this is to test the individual got infected or not. And usually it's infected and recovered. So when you got the antibody. So it used to believe if you got the antibody, you are safe. You don't get infected again. But it seems at the moment there is a report for people still get affected again. So it's basically this virus is very clever, or we know very little about this virus. So we are talking about the nucleic acid test, okay, to the more accurate test, and to test the individual whether got infections, whether got a virus and the infections or not. So this is what it is currently what is done. So you got to take a swab. This is a lot of this in the news in the car park, in the test stations, basically take a swab, and this sample, this will send to the lab. And when you got the lab, it's quite complicated. They usually do an RNA extraction and then run the RT-PCR. This polymerase chain reaction amplifies the virus RNA. So then you can get the detection, easy to detect it. This PCR, you take at least 90 minutes. And then you have the time to do RNA extraction. And then the time for transport, that's the main thing, because you have to get the transport to the lab. And then you see that there's a big problem at the moment, is a shortage of reagent, because every country all do the PCR test. Then the reagent supply become a big issue now. But overall, this process is slow and expensive, right? So if you do a private test in London, it costs you £300. So it's, and I think at the moment, some cheapest commercial companies offer like £50 to do a test. So what is our test? So our test is much simpler. It's only 3 steps. So it's you basically, we at the moment we can use swabs. So you got the swab solution. We directly add into our test tube. Our test tube is actually a small test tube here. It's very small. I think if I can, I will show you later. And very small. And you just one step, heating up and keep it at the 65 degrees centigrade. Okay, 65 degrees centigrade. And for 30 minutes. And quite often we got the results in 15 minutes or less. But then if you got a negative results, make sure it's a true negative. So we ask people to do 30 minutes, okay? And then you just look at the color change. If initially pink color, if it's still pink, that's a negative, no virus. If it turns into yellow, that is a positive, okay? To heating 65 degrees, you can do any different ways to do that. You can use heating block, you can use oven, you can use thermal flask. Like we said, you just use hot water to make it 65 degrees and heating salts, that's it. Because the tube is so small. So the features of our test is fast, is end-to-end, 15 to 40 minutes, longest is 40 minutes. And you don't need any equipment, and it can do it on the spot. So it's a true POCT, point of care test. And it's very simple. We don't need to do processing of samples. We can use the swabs, and we can also use the saliva and the mouthwash. And easy to operate, just heating up. And we call it directly results display. You just look at the color change. And it's very sensitive, and it only detects this virus. And another important contribution we made is that we get rid of the cold chain. We put all the reagent in the test tube and dried. So you can actually put it in an envelope to send it to anywhere in the world without cold chain. And this is in comparison with PCR test. The PCR test kits, at the moment, most of them, almost all of them, have to use cold chain supply, transport and storage. So we don't try to replace central laboratories for test with this like a decentralized testing. So like in GP clinic, airport, nursing home, schools, universities, and then now at the moment like in the sports events, all this type of sports events. Important thing, of course, is whether it works or not, right? You said, is all great, it works or not. So basically, we proved it works. We actually validated in University of Oxford, the John Radcliffe Hospital, and our collaborator is Dr. Monica Anderson. And it has been, I have to say, actually, one of the very important difficulties we face is to get clinical validation. You see, there are so many people say that we have kids, we have a test, but then is actually when you got the clinical setting, a lot of them is not practical. And ours, we started very, very simple as possible. And all the consumables are readily available because we want to make it simple and we want to make cheap, make non-expensive. And then this actually makes the medic really, really exciting. So we actually compare with actually clinical validation, directly use swab samples, and our results is comparable with all the standard PCR, and it's much faster. And it's only test this virus. This is just a show. It's only test this virus. It doesn't test. It does not react with any other virus. And this is more technical now. Is say how sensitive it is. So we can reliably detect 10 copies per reaction. But actually, in a more precise setting, we can detect 1 copy. That means if you got a virus, we can detect it. Okay, so I was very, very good, but then that needs a better instrumentation. So this is actually very good results. We actually can compare with any other technology. We now can claim it's just as good as a PCR. So then the question is that when you test the patient, test the people, quite often, so we said that, can you get guarantee 100%? Well, the question is that, I think the answer is no, and nobody can, because there's 2 problems here. One is the errors in sampling. You see, actually there is a report published that show the sampling method make a big difference to the results outcome. Depends on who sample it, what the time to sample it, and the way you see, and that's actually the single biggest error. And there's a worst scenario at the moment, the report in PCR test, standard PCR test, the accuracy is only 30%, okay? So it's really a lot of factors filtered there. But for us, and we put about, we put this line here, about this is all the virus loading of patient at different time, basically say we catch all of them, okay? So what do we miss? We miss people almost recovered. If you are staying in hospital, almost recovered, but you do a PCR test, they may still be positive. But then they are not infectious at all, but they are still tested positive. So that type of patient, we sometimes miss that. So our tests generate a lot of interest. So basically, we actually get the major like media, CNN, BBC, a lot of newspapers, and reported this. So it's reported over 14 countries. We scientifically, we published 2 papers. We have another one in review. We filed 2 patents. And so it's all actually generate a lot of publicity. It's just, I think we are lucky. And this is because the test is always on the spotlight. We are just lucky ourselves on this. So I will show you a little. This is a television program made, I think, by a Global China Nation Network in London. And this actually, this is their little clip. I think that's answer some questions better than I explain it. If it's turning to yellow, because that's turning yellow, that's a positive result. So that means it's get virus. With the still like.
Professor Zhanfeng wants to release a test anyone can use at home to find out if they've been infected with COVID-19. He's been working on this since the outbreak in Wuhan in China.
It's a test the individual whether get infected and still infectious. So if I'm tested positive, that means I got the virus, I can spread the virus. I should go to isolation.
And this is how the test works. You take a swab sample, add some solution, shake it, add some more solution, put it in a test kit, another drop, and the solution becomes pink. It's then heated up to 65 degrees, either using a hot block like this or hot water. Leave it for 30 minutes and it's time for the results. The beauty of this test is in its simplicity. If the colour changes from pink to yellow, it's positive. If it stays pink though, it's negative. And the team here are hoping that eventually they can make this test so simple that we do it ourselves and at home. For now, Public Health England is testing healthcare workers at drive-through centres by taking a swab test. The samples are sent to a lab, and with so many tests to process, it can take up to 2 days to get a result. These scientists aim to release a quick test that can be carried out at clinics, pharmacies, even airports, and at home. Oxford University has made this project a joint collaboration with China. The university has a branch at Suzhou Industrial Park in southeast China, and half the team is working there.
It means we're able to react very quickly, and also key for us has been when the lockdown in China was happening, we could work here, and now that everything's switched around, we've got the ability there. We're less affected by impacts in transport, and we're able to make the best use of patient samples, hospitals and manufacturers worldwide.
And the interest in this test is global.
We like to issue like a royalty-free license under to manufacturers all over the world. It's non-exclusive. We had interest expressed from 14 countries so far.
In clinical trials in China, the test had a 100% rate of accuracy. It now has to replicate that in the UK. If it does, people here are one step closer to community-wide testing and ultimately getting back to enjoying life outside. Nicole Johnston, CGTN, Oxford.
Okay, I think that is a little clip, and then that was done a couple months ago already. Time flies. And then our work, as Professor Guo suggested, and I think he's very active in the Royal Academy of Engineering, and we got this President Special Award for this pandemic service. And this actually was a big boost to the young people, the team members. This is not to me, it's to the team. And because actually they dropped everything, worked very, very hard from late January, and day and night. And quite often we had group meetings 8 o'clock in the evening. And then during the day, they're all in the lab. So they carry these 2 lads, carry these big bottles. This is all field experiment, a lot of experiment, because they have to do a lot of testing. And they just show how much work they have done. It's a big boost to their morale. Because everybody working on volunteers. You see, this is all student postdoc, and then nobody forced us to do this. It's all volunteer service. And when we got the test, we said we needed to actually apply for the regulatory approval. In the UK, it's MHRA. And they said that you are a university, you cannot apply this. Your university is not a commercial organization. So the university, the Oxford University Innovation and Technology Transfer Fund arm, they quickly launched a company. They formed a company for us called Oxsed and Limited. So this is actually, we launched a company without any investment. So we have an entity and it's social venture company, and particularly try to launch this test case. So we formed the company, then we we're working with Oxford MISTA, that's a medical device company. We got the CE mark in July. And so we are ready, then we can launch the products. So it's actually very quickly, we from R&D, product development, form the company and launch the products, okay? So it's very, very quickly we got products, we can get the products. So this is like the timeline. If you think a lot of things happened when in China in January. In China, we started later January when the Wuhan lockdown period, and then all the activity in China lockdown, we all done this research R&D in Oxford. And then later, UK, later March, UK become lockdown. But then our group, because we're working on the test case, we all had access to the labs. So we never had a lockdown, but we just watching ourselves. But the difficulty at that time is that there's nowhere to get food, nowhere to get drink. So this is actually the team worked really hard. So that's why we got, you see, this is timeline. So it's very, very quick. And it's a very special case. So we form a company, we got the products. And we now get to commercialization step. So we got the CE mark, we are launching the products, as I said. So at the moment, we can supply 100,000 tests per week this month. And next month, we can reach to 1 million per month if we got orders. Again, at the moment, the company does not have single employee. It's just all our team still keep it floating. And of course, the next stage, we are looking for investment to try to get it operated in a pure commercial sense. So the products at the moment, they evaluated in the USA, in South Africa, India, China, Saudi Arabia for regulatory approval. So several countries to doing that. So because this started with China, so we licensed to Sinopharm in China. And this is a new, they announced this because they have been working since, well, we licensed this some time ago, but we did all the technology transfer. Now they can do manufacture, they can do large scale manufacture now. So they made this announcement. China Bio is part of Sinopharm, and they are the one to do the vaccines, also to do the PCR test, one of the first company to get the PCR test kits. So now we transferred the technology to them, they licensed it, and now they can make large scale production in China. And they just announced that a couple of days ago. Now to Hong Kong, we are working with a company called Prenetics. And this company they have a presence in the UK. They are the one to test Premier League football clubs at the moment, okay? So they test the footballers, and they approached us. They said, okay, we heard of this case. Can we actually evaluate your case, test with the football club? And then, so they are the one to do, they also want to get this to Hong Kong. Actually, I was told is get approval from Hong Kong authority, which I don't know what the exact details, but this is from the Prenetics as what you want to do basically is for large scale, low cost, frequent screening. Now, if you think about it, how we deal with this virus at the moment, you see vaccines still have some time to go, although the Oxford vaccine resumed clinical test. Okay, it's basically had a pause for a couple of days and now resumed, but then it's still some time. So the moment how we get back to normal life, we really need a frequent testing. You see, how frequent? Probably I would say once a week, probably, or even more. But then you really need a low cost of testing. If you actually, you see, otherwise we'll bankrupt any nations, any countries. So it's a need a low cost, frequent cost, and then we need to decentralize. We don't need to have to collect all the samples to the lab. So this is actually in the clinic, in the airport, in schools, and in the UK, one of the big issues facing the society is the schools, because the people worry about the school, the children, they are fine, but they might bring the virus back. So the Prenetics colleagues did a comparison between our test and the PCR, antibody, antigen. And I borrowed these slides. I said, can you give me some slides? You do your work. And basically, this is their bit. They said, you see, and you tick out the box. I think, you see, if don't tick the box, probably don't put it there. I think, so this is all compared to other tests. Our test is actually quite good. I was told this already get approval in Hong Kong. And I have to, I just got this today. So one of the important things we like to push is point of care test. Okay, it's not actually just collect the samples, send to the lab. We are not competing with central lab, but it's adding on, because to get that really test the capacity. So we are promoting, for example, test this in the pharmacy. So in the pharmacy, and then we did a calculation, you only need £500 to set up a test hub, and you can test 200 people a day. So if we got all the pharmacies used, and some, you see, we can set this up in schools, in private clubs, in football clubs, in all the sports events, and like mobile test stations. So it's very straightforward. You see, you take the sample, you either use swab as a standard one, or we promote a mouthwash, basically you gargling water, and then because that will collect not just the water in the mouth, also behind the throat, that area. And then our saliva. So from the sampling, you collect the sample, you get the sample, then the first thing you do is inactivate the virus. Okay, in case it's got infectious virus in it, it's positive, then you just inactivate it first. You're heating it up 95 degrees for 5 minutes. That's very straightforward. You inactivate the potential virus, so safe to handle. Then you can open the test tube because you see inactivated. Then you take 25 microliters of liquid, just a big drop, into our test solution in the test tube, and heat it up. So that is very simple. Look at the color change. Then you can use the mobile phone apps to collect the data, transport the data. So that's like a point of care test. And also this can be done entirely. We actually quite ideally use the home test, self-test. But actually, but the government, UK government, and the US government as well at the moment, they don't allow true home test. When they say home test, they collect samples. They take samples away to do a test in the lab. They don't allow you, they don't promote to do it at home yourself. The reason is that they want to have the test and the trace. They want to trace people. But if you test at home, then you may not report. So we can do a self-swab sample. It's actually a lot of YouTube. Actually, the UK government at the moment, they send the swab tube to the home, send the link to the YouTube to see how you collect the sample, and then you send this back. So self-sampling is possible, but it's not very uncomfortable, okay? And the mouthwash, that's what we are promoting. Basically, just gargling this, and we can collect that. It's very much easier. But then you just, because you are tested at home, you don't need to worry about infect other people. It's all in your household. So we can directly use the sample, adding to our test tube. You put it into you make a 65-degree hot water in a thermal flask, okay? You either measure that or you just mix like 3 parts of boiling water, 2 parts of cold table water. That's mixed roughly about 65, 67. That's good enough because we don't have to be exactly 65. And then you just wait for 30 minutes, then you got the results. So this, you don't need any equipment and you can just do it at home. It's a pity at the moment, they're not allowed to do home self-test. But sooner or later, this will be a norm. I think people want to do home test at home. So this is what I see. I say the game changer, and we may have to change this. So first thing in the morning, you wake up, instead of brush your teeth, and then you actually gargling, use a small mouthful of water. Okay, adding 1 drop of the gargling water to the test tube, put the test tube in the thermal flask, 65-degree water. People said you don't mix it, you can buy a thermal cup, and there's various devices you can set the temperature at home, and very easy. I said, fine, we just need 65 degrees. Then you can go to shower or breakfast. Then when you come back, take the tubes out, just look at the color. If it's still pink, you can go to work, you're safe. You don't infect other people. If yellow, just say, I stay home, I'm on isolation. I think that would be the way I see it. Now, so I will put the last slide. This is actually the slogan of OSCA, is the future of science is global. And Oxford, we actually truly believe international global is the way forward. International collaboration is very important. And so I think with that, I say thank you and stay safe. But I like to, do I have time? I'd like to give you 1 minute demonstration how this can be done, right? Because I said that you said this is simple, and how simple it is. I'm at the guest bedroom, so you can see me, right? So this is our product. So unit get 10. This is actually the product unit. Okay. So take one out. I take one out. This one already reacted. I need this is not the. You will see why I already get a. This is anyway. I show you just to take one of this, but this one is already reacted used. I was not given one. They look like a pink. They should look like this. Okay. Right. So you take one of this. You got the thermal flask. So thermal flask, you put some water in it. Okay, you got hot water, 65 degrees. I didn't prepare it. I just want to show you. If in the kitchen, you can use your kitchen temperature. This is for cooking, everything, 65. So what do? Get the water ready. So you can get the water. I just want to show you this indeed water is not a magic solution. I open it up. You just collect your gargling water. You can provide this kind of pipette, very little one. So if I open this, little test tube. Okay, this is opened. And then I just take this, you mix that. Okay, I was doing this now. Okay, take this out. You put, this is the one, I put 1 drop. Okay, 1 drop in it. I now seal it, adding to this, and then close it. Keep 65 degrees hot water for 30 minutes. When you come back, you're looking at, if it's pink, like this is a negative, clear. If it turns to yellow, that's bad. If I put it here, well, you can see pink and yellow. So pink is negative, yellow is positive. Positive got a virus, pink is good, yellow is bad. If you do a test like this, I hope you all pink. Thank you.
Thank you. All right,
okay.
Thank you very much, Professor Cui. Please stay here, maybe you'll take some questions. First of all, congratulations for your great achievement, and congratulations to your team. And it's truly remarkable. All right, it's only 7-8 months, right? A lot of people are really stay home and feel sorrow and feel hopeless. But your team is working so hard and find the solutions for the mankind. It's really great. Congratulations. Now, I think now we take some questions from the audience. How this be done, Meta? This is people will ask randomly or you raise a hand?
Ika, if you could please ask people to use the Q&A function at the bottom of their screen. then you will get their written questions, which you can then select and read out.
All right. Okay. So I don't have the question yet.
Okay, so just call for some questions.
Yes, okay, the audience, if you have questions, please type in this Q&A sessions, and I will read out. If it's not, then we will ask question. Okay, so may I ask the first question then? So Professor Cui, so certainly this is very useful for the, you know, the point of care testing, but I think one of the very big usage is really for the developing country. And I think it's in a developing country, you know, you don't have the full capacity to do the lab-based testing. And what's your plan to actually roll out to the developing countries, something like in the Africas or either probably in the Latin America? Now Brazil is doing so badly. So what do you think about this? And what's your plan?
Well, the plan is that we need collaborators, okay? We need collaborators, local ones, because actually we ourselves, we don't have financial, we don't have means to do all the marketing and reach that. So we are doing a couple of things. One is actually we are working with the UK Commonwealth Office to see some, through their connections, whether we can actually channel this, basically to get the local people know this does exist, right? It's relatively cheap, and then is you don't need the equipment, you can do the test at, you see, on the spot. And so that's one channel we are working. We are working with the Commonwealth Office, and then through various channels like charities, we actually try to alert like Gate Foundation, this type of charities, and say that this can, whether their channel can help on this. So we for example in India, we are working with we are teamed up with some Indian companies with their testing and the distribution network. And we try to working with them. And basically we are looking for partners in South America as well. We have got 14 countries, different countries. They're not just developed countries, right, and many of them middle low income countries. But we just need the partners, local partners.
Yeah. But follow this question, right? Now I think you formed a commercial company, but also your model is licensing, freely, without charge license fee, and which is great. Morally, this is really the high ground. But however, from commercial model point of view, how the company can make money? If you,
I think yes. Good questions. Actually, the company does not because actually it's a social venture company. The university does not charge us license fee as well, but this is only limited to the pandemic period. So our hope is that we don't make much money during this time, but after the pandemic, then we will start to recover some of the costs.
Right. You may developing some vaccines, similar methods for different disease. That probably, that's probably a different idea. Okay, now I get it. I get it. But in this way, I think that one of the very big application area is the school opening, right? Yeah. Hong Kong is now very cautious, extremely cautious about the school opening. And really, I can think about this is, how do you think this can apply to the city like Hong Kong, which is extremely cautious, but also have a limited number of university, because they have the history of SARS. So they have very sensitive about the situation, but they actually did a very good job on the control the pandemic. But now, would it be possible to collaborate with the Hong Kong government to actually first time apply your kids to actually working for the school opening? This will help a lot of people.
Well, I think absolutely, we would love to, because actually we actually planned to do a testing of 1,000 student school, a school with 1,000 pupils in the UK. It's all planned, and there's a delay because of legal issues from the school. They said, look, we have to the question they asked us, they said that if children tested actually negative, but then bring the virus back to the family, infected the grandmother, so whose responsibility of that? You see, this type of thing. Now, we actually worked out the test school, okay? So we have test station, because actually what the ideal scenario is at least you test all the children twice a week. So what you do is not, you don't use swabs, because the swab, nobody likes this put in the nose, right? So you use the mouthwash. And then the other thing is that you don't test everyone with a single test case. You pool the samples. In one class, you say, okay, you can have 20 pupils. You take the mouthwash and you mix their samples. You do a test. Then this way, if it's negative, this 20 is fine. It's like a bubble. Like the UK, they use the word bubble. So this bubble is fine. If it test positive, you try to identify who got the virus. But if you got a positive, you need to isolate them anyway, all of them in a bubble. That's the UK policy. So you don't test everybody individually. You test them in a group. But you test all of them. And so in this way, the cost will be done, because if you test the 20 in one go, your cost is a fraction of 1 in 20. So you can repeat. That's right. You can repeat the test. But the important thing is take frequently repeated the test.
Okay. So there's one question for you. Congratulations, Professor Trey. Great test too. Will the test kits be sold in the general drugstore for public?
We are trying to, that's the objective. But then at the moment, because they are not the UK government, our approved test is for non-personal use, is for professional use, for trained person to operate. So it will not be at home, you do it yourself, like I showed you at the moment. And then, so we actually want to put in the drugstore, and we like to persuade the drugstore to set up a test station. So the individual can bring the sample back there to test on that spot.
Right. I think your idea of a poll test will be pretty practical, right, for university. We may actually, have you had, sort of active dialogue with Hong Kong government or somebody? So we can actually really think about it to do this one for the school opening.
I think it's because that's the only way you can test everybody every week.
You can
Otherwise,
test every week.
You bankrupt the country.
Yes. Poll test is really possible, right? You can put 50 people in one bunch, right? And the test is isolated. That should be quite cheap. Particularly Hong Kong, right? The intensity is quite low. So therefore, we can actually do a very good job. Keep the campus safe, and we can resume completely our online, our face-to-face lecturing.
I think in the UK, for example, all the university opening this month and next month, this is a big issue, you see.
Yeah, it's a very big issue. And Meta actually raised this question, so I answer you. We may actually talk about this actively. So who's your Hong Kong collaborator, this company you mentioned, right?
No, at the moment, this company called the Prenetics. I could put you in contact with them because
probably.
Because the only reason they approached us is because they are doing they have a branch in the UK. This branch is doing the Premier League football club testing.
Right, I see.
That's why.
Yeah, I think we probably try to contact the government to actually, if the tools actually got validated, approved, then we can certainly use it. That would be very good for the university.
Yeah, I can do the introduction, or you can find the website anyway.
We do that. Okay. Yeah. So another question for you is one from the evil one, is one of our staff, and she mentioned about your team is very bright and diligent, but keeping them working long hour from it cannot last forever. So what's your plan to transfer and spread this knowledge and technology, which you already mentioned? So basically. How can you scale up your activity? You just give it away for other company, for collaborator, right?
Yes, I think we need the partners. So yes, yeah, we are very open for collaboration for people at any part of the world. You see.
Okay. Thank you very much, Professor Choi. A wonderful talk. Congratulations again. Okay. Hope we come to Hong Kong very soon, and then we can work together.
All right. Thank you.
Thank you.
Bye-bye.
Bye-bye. Bye-bye.
Bye-bye.
Bye.